I'm sure everybody can hear me. Skok Skuman speaking. If I can just do some housekeeping rules. If all of you that have tuned in can maybe just mute your microphones, so that we don't listen to everyone, it would be appreciated. And thank you for joining us. Firstly, I think it's important. We are planning to host a series of these talks, in order to get a bit more information out to you guys. We are going to kick off the afternoon's festivities with Gerda. Gerda is going to tell us a bit about the Boers. It's an interesting one. It is slowly becoming more of an issue, not just for South Africa, but globally. Speak to other growers organizations in other parts of the world, they are noticing similar trends. So, yeah, what we're going to do, I think let's reserve the questions till after the time. We'll provide some time for Gerda to react, and if you don't want to raise your hand, there's always the function that you can type in a question, and we'll allow time for that. It looks like the numbers have stabilized. Without further ado, Gerda, can I give the baton over to you? The floor is yours. Many thanks.
Yes, thank you, Skulk. Welcome, everybody. I've asked Alria to help me share the slides. It seems from my side there's a little bit of an issue. So thank you, Alria, for sharing the slides. And
sorry,
apologies for the issue. Yes.
Sorry, can I just, would you like me to share the PowerPoint version or the PDF version?
Well, if you can go per page, because now if you can make it a little bit larger so that I can see the full page per slide, I think the PDF version is easier. Otherwise, I'm going to ask you too many times to go to the next one.
Okay, let me just share that one again.
And like I said to everybody else, apologies for something being wrong with the slides. But yes, I'm here today to give you a brief update on ambrosia beetles and their fungal symbionts. Thanks, Alria. You can go to the next slide. The next slide is just a little bit of a background of what are ambrosia beetles. It's important to understand that they are wood-boring insects. When you look at them in their native range, they typically cause minimal damage. That is because they will attack stress trees, and oftentimes they have natural enemies that will keep their numbers down. This is then different when they are in their invasive or when they invade in their non-native range. So we need to understand that they can then be problematic. They have an interesting symbiotic relationship with fungi, and important when we think about these fungal symbionts, they can also sometimes be a problem, especially in their non-native range. So Aria, you can go to the next slide, thanks. A little bit more about understanding the life cycle or the host beetle interaction, and that is when I mean something that was introduced into the country. So I'm trying to display here a macadamia tree that undergoes a stress. The stress can be an extreme environmental condition, a period of heat or drought. Stress can be poor cultural practices, or it can actually be attack of pests and pathogens. But when a tree is under stress, it actually emits these mostly alcoholic volatiles. And although the stress can actually be temporary, it's not necessarily detrimental to the tree. These sort of volatiles that the tree will emit actually attract your beetle to the tree, and that can then allow the beetle to sort of invade and to really start breeding and establishing in your tree. Okay, so Aria, you can go to the next slide. And so because we've seen over time a sort of an increase of these beetles and these borers in our orchards in macadamias, we partnered up with CropWatch Africa, and we actually set aside 24 trapping stations, both in KwaZulu-Natal as well as Mpumalanga. And so the slide just represents where we set out those traps. Aria, next slide, thank you. Little bit more background for these specific traps. We were interested in really capturing the polyphagous shuttle borer. Later in the presentation, I'm going to talk about the shuttle borer again. But then in addition to this, we were interested in any other beetle. And for that, we set out ethanol traps. If you think back, or we have talked about this sort of beetle and the host interaction, we know that when trees are stressed, they emit these ethanol compounds, and that is what can attract the beetle. So we wanted to see, can we capture the polyphagous shuttle borer? But in addition to this, what else can we capture? Okay, next slide, Aria. And then, of course, thereafter, we went to say, we identified, we started with morphology, but thereafter we did a DNA sequence that was then the COX-1 gene. Thank you, Aria. So the following, it will be then be 3 slides. It's a very short summary of what we found over the 2 years. In total, this represents close to 15,000 beetles. If anybody is interested in more detailed reports on the beetles and their presence, you are welcome to send me an email. On this slide, the first 2 bars, first of all, the left one would be 2020, the one to the right will be 2023. But the first 2 beetles that I'm trying to highlight is, first of all, beetles that we found within the Bostrichidae family. These are not beetles that we should be concerned about. These are beetles that are actually attack sort of products that are made of wood. The second one is then Hypothalamus humpii. Yes, the name might be familiar. We know in Australia about the tropical nut borer that attacks nuts. But in this instance, I'm still talking about borers that were part of, found inside sort of trees. And these are typically ones that we shouldn't be concerned about. Okay, so then next slide, Aria. Some of our beetle of interest is then Pronovius, Caffiprenus, Xylosandrus crassicaeusculus, some Xyloborus species, as well as the Ambrodioseum. And these are beetles that we typically see in high numbers, and perhaps beetles that we should take a closer lookout for. Okay, next slide, Aria. Then, of course, I want to highlight the polyphagous shuttle borer again. I think if we look at something that might be alarming, if we look at the 2022 and 2023 survey, we do see an increase in the number of polyphagous shuttle borer that we then obtain from these traps in either Mpumalanga and or KwaZulu-Natal. Next slide, please. So what I'm trying to talk about here is again the polyphagous shuttle borer. Some of the first work that we've done is we've confirmed that it is a macadamia is a non-reproductive host. And so in terms of this, macadamia growers should not be so concerned about this beetle. However, what we've seen happened in pecans is when the tree really undergoes stress, you can sometimes see that there's a physiological change. And what we've now seen in pecan is that some pecan trees have now switched from, first of all, being a non-reproductive host to a reproductive host. So this then means that not only will you be able to isolate the beetle from the wood, the beetle actually can reproduce and the larvae can actually stay in the wood until adulthood. And so this whole idea, sorry, I'm not done, almost done yet. The whole idea of it being non-reproductive or reproductive, specifically for the polyphagous shuttle borer, is strictly linked to its fungal symbiont. The beetle needs the symbiont. It cannot complete its life cycle without the symbiont. It literally feeds on the symbiont. And so the better the symbiont can grow in the wood, the better the beetle can grow. And where we found in both max and pecans initially, the Fusarium symbiont cannot grow very well, we've now seen in very stressed pecan trees that that symbiont is actually growing quite well. And so when we think about the other beetles, it's also important for us to go and look at their fungal symbionts. So next slide, Aria. So this is then our aim for this year. We have listed which is the most common beetles that we see present in macadamia orchards. That will then be the ones that are not the polyphagous shuttle borer. We are going to include Euplatypus parallelus It wasn't picked up in our traps, but we know it's present. And we're going to look at what those fungal symbionts are. We want to identify them. But in the end, it's important for us to see, but how well will they grow in macadamia branches? Because it's a combination of the beetle boring into the wood, but the fungal symbiont then being able to really grow quite well, and that the combination of the success of the fungal symbiont and the beetle can then really cause damage. Next slide, please, Ariel. So it's difficult to really talk about economic impact, but when I think about the impact, and something that is of concern is that perhaps you will be in Mpumalanga and you will see a tree that is infested by macadamia felted cosset, and you might have a strategy to really control the cosset. But because the tree is under stress, you will often see sort of the ambrosia beetles. Of course, it carries this fungi symbiont, is attracted to it. And I think the combination of the beetles with scales can really be problematic, and I think it can result in tree death. Some examples that we've seen in Mpumalanga is you typically also start to see quite a lot of beetle attack if it is an orchard with a little bit of a root rot, Phytophthora root rot problem. And so it's about, yes, there's a Phytophthora problem, but now you need to be concerned about both the Phytophthora and the beetle. And so the economic impact, or I think resulting of tree death, can actually happen in macadamia orchards. Next slide, please, Ariel. So what can we do about it? Our first management option for beetles, unfortunately, is just we need to prevent this as much as possible. We need to try and keep the numbers down. What we would recommend is then solarization of then branches that are infested or highly infested trees. Next, Ariel. Not specifically work done by myself, but done by various people in FABI as well as in Stellenbosch. We unfortunately know that our chemical options are very, very limited. This is just an example of a tree, tree injections, but there's also this sort of level of phytotoxicity for the tree. So I think once these populations established, there's really no chemical option. Ariel, next slide. Where we do think are some positives is we've tested some commercial entomopathogenic fungi. We've looked at commercial products containing trichoderma. Of course, the trichoderma would be for the fungal symbiont. The EPS will be for the beetle. And it seems to work really, really well. The biggest issue is how do we really deliver the product to the actual beetle? How do we get the product into the larval tunnel? And again, not work that I'm involved with, but there's an exciting project starting at Stellenbosch where they are going to looking at mites and the probability and the ability of these mites to carry these projects, to carry these commercial products. Our last thing, thank you, Alria. You can go to the next slide. Is then we always think in terms of classical biocontrol. If you think about macadamia felted cosset, I had a quick slide there. The ARC is looking into bringing in a parasitoid from its native range. This is just a picture of Brett, Vilma, and others from FABI that visited Taiwan last year. They are in the process of looking for a natural predator specifically for the polyphagous shuttle borer. And so I think in terms of shuttle borer release, we hope that in the next few years, this is something that we can commercially release, at least for shuttle borer. This can be really help us a lot. And so my last slide is just an acknowledgement, of course, CropWatch for the 2-year survey. They helped us a lot. Of course, SAMAC for funding, and a list of the students that helped with the beetle surveys, and of course, then the macadamia growers that allowed us to sample on their farm. Thank you very much. And again, apologies for my presentation issue. I'm happy to take questions.
Yeah, any questions for Gerda?
Alria, I see you have a hand.
I was just wondering, Gerda, do you think we need to repeat our pathogenicity trials with the shuttle borers fungal symbionts in a similar way that Angel did her work with Dieback, so on stress trees, some or other form of stress?
Alria, thank you for the question. It's actually something we talked about. that, you know, we want to see is it also going to happen in macadamia? So if there is a really stressed tree, can polyphagous shuttle borer start reproducing? The problem is for polyphagous shuttle borer pathogenicity trials is you really need to use larger branches. So the seedlings are not going to work. And then if you want to place those branches under stress, we have to come up with a good plan on taking the big branches and putting them under stress conditions. But yes, it's something we talked about. I think it's something worth investigating. I think the practicality of cutting off a branch and trying to keep the branch sort of alive long enough to do the path trial and then to induce a stress might be complicated. But with maybe talking with you and Skalk and the R&D committee, we can brainstorm on how would you induce that stress. But I think it's worth doing, yes.
Gerda, maybe from my side, a question I've always wondered, and while I'm having you here, I'm going to ask. It is, you talk about solarization. Will the adult beetles not fly away if you solarize the stems? We've done similar work on the coffee stem borer a long time ago, and these are absolutely amazing insects. So just a question. I can understand that you know, you'd probably kill the immatures if the temperature in the stump goes up too high, and if you leave it in the sun for long enough. But the adults, won't you simply increase the spread of the insect by doing that?
I think you might. It depends on whether or not they can actually chew their way out of your plastic sheet that you cover it with. So that would be something that you need to look for. And I think if you want to do it, look for, can you see like small beetle holes in that big plastic sheet that you cover the tree stumps with? This was from, maybe we can have a better discussion with Hadeseptoe from SAPA, where at some point, I think burning is, you know, burning would not be an option. The other one would be chipping, but you really need to go really small. So chipping is almost not something that they would recommend. And if you talk to Hadeseptoe, I think he's from the Pecan Growers Association. The picture is also from Pecan Orchard that they solarized, underwent solarization. Out of the current 3 options, his recommendation is that it is the best, but none of it is actually foolproof. I think also you would get a lot of escapees if you try to really burn it, and I don't think you want to make such a big fire on your farm. And then the size of how small you really need to chip them, apparently it's also almost not feasible, if I'm making any sense.
No, you make a lot of sense, Gerda. It actually, for me, is rather important that we dwell on this subject and we understand. Because on coffee, we tied the trees with mosquito galls, and the insects were able to eat through it. But that's a different insect. So I think this is a very important one that you know that we will have to look at in future. And to be sure, no, this from our side, I always wondered, and but you answered it very well, and I had your answer in the back of my head. You just confirmed what I wanted to know. Ria, is that an old hand or is it?
I think it's a new hand.
It's a new hand. I was wondering, Skalk, if you can maybe just explain to everyone online what the project on the phoretic mites is. So Gerda mentioned in her presentation, so we have a joint project with SAGA, so it's the Avocado Growers Association, Hort Grow, and the Pecan Growers Association on using these phoretic mites to introduce pathogenic or control agents for the pathogenic fungi into the tree. Maybe just 2 sentences on that project that's just started.
Yeah, it's a good one. You know, with researchers, we always like big words. Phoretic basically just mean that it hitches a ride with another insect. If you look at the holes that these insects make make in a stem, it is tiny. And if you look at it will be physically impossible to spray a chemical so that you get residues actually going into the hole, unless you go from hole to hole. So that means that chemical control is to a large extent not feasible. So we'll have to look at something else. And phoretic mites is basically mites living inside the tunnels, that is created by these beetle borers. And what these guys are doing is to inoculate, or the idea will be to inoculate these mites with pathogens that will be able to. You must remember, when you look at a pathogen, it has two phases. It can either grow as a saprophyte or a parasite. Saprophyte, when it ends up on organic material, dead organic or living organic material, and a parasite when it ends up on insect chitin. So what they do is to inoculate the mites with a pathogen. The mites carry it into the tunnels. Inside the tunnels, it's moist, it's full of nutrients. The spores germinate, grow saprophytically, and once it comes in contact with one of the insects, it starts growing parasitically and then kills these beetles. That is the theory. In practice, it's a very strong team, and if there ever was a chance of making it work, we'll probably get this team to do that. I'm quite upbeat about this process. It is probably the best chance we have at the moment of controlling these beetles. Does that answer your question, Elria?
Yes, thanks, Kalk.
It's a long way of describing, but it is a complicated process. I don't see any other hands. If there are no more questions, I would like to introduce the second speaker of the afternoon, Fahimi Fahimi Jami from ARC in Pretoria. Fahimi is looking at branch dieback and the diseases involved there. And when one spends some time in the orchards, the one thing that I tend to notice is that the frequency of trees infected with branch dieback is most certainly on the increase. And so this is a really, really important project for the industry at the moment. So Fahimi, we are in your capable hands, and we're looking forward to what you're going to have to say to us.
Great, thank you so much. Fahimi, sorry, it's Gerda. I did apologize before the time. Thank you, everybody. I am going to sign off. Fahimi, once again, sorry for missing your talk. I have an unexpected need to quickly get my child to a doctor's appointment. But if there's any questions, email me. Fahimi, once again, sorry for signing off and for missing your talk. And goodbye, and thank you, everyone.
Bye.
All the best, Gerda.
Okay, thanks, Scott, for an introduction, and good afternoon, everyone. Some of you maybe you have heard before about my project, but I'm gonna I have updated, and I will take you through. So for this project, we have a few objectives. I'm not going to read one by one here. I'll take you through one by one through the presentation. So we had, as most of you know, we have a loss of diebacks on macadamia trees in the orchards. So first step was that we identify what is causing the dieback. For that, we isolated from 11 different farms in KwaZulu-Natal and in Mpumalanga. From the trees that, as you see in picture, they are showing there either one dieback on the trees or the tree is almost dying. So the samples were collected, and when we cut the main stem, you can see this discoloration. We could see this discoloration in the main stem, and fungi was very aggressive. Even 3 hours after collecting the samples, the fungi, as you can see, these white hyphae start growing. So I isolated them and purified them and identified them based on molecular analysis, which we identify 9 species, 7 Botrytis species, and 2 Dioportaceae. which 4 species, as you can see here, they were common in both KwaZulu-Natal and Mpumalanga, while the other species either they were in Mpumalanga or they were on KwaZulu-Natal. So from here, we took these 4 common species for a next step, which it was evaluate which one is more virulent and if we can find any tolerant macadamia varieties against that. First step for our testing virulence, we tested on the trees in the greenhouses. And we tested on 450 trees. If you are interested to see that how we tested, first we made a wound on the stem, and this is the fungi that we get from the petri dishes. We put it here and then wrap it with paraffin to make the moisture around it. And then after 6 weeks, we remove the bark and measure the lesion that the fungi made it on the trees. So this is the results of the length of the lesion of those 4 species that they were common. One species, Lasidioplodia theobroma, was significantly to produce significantly longest lesion, which actually in Lasidioplodia theobroma was the most common isolate. 80% of our isolates, they were this one. So then we took that isolate, did that species to test it against these 3 macadamia varieties that mainly are common in orchards, Beaumont, A4, and Nelmac 2. The process was the same. Again, we inoculated on the different trees, and then after 6 weeks, we removed the bark and measured the lesion. So 2 macadamia variety, A4 and Nelmac, there are 2 days significantly produced shorter lesion in compared to Beaumont. And you can see in the picture, this is the Beaumont one, and this one is Nelmac, and this is control. So we wanted to evaluate if there is we know what fungi it is causing dieback, and now we want to know if there is any fungicide it can be effective. We chose 14 different fungicides from Syngenta and Bayer's, and we test them first in the lab. All 14 fungicides, we tested for 4 concentration of each one with 10 isolate and 10 3 replication, and plus control, we ended up with 1,800 petri dishes. So in this picture, this is what we did in the lab. In these petri dishes, we use the fungicide, and then we inoculate the fungi here, there, and this petri dish does not have any fungicide. You can see that the fungi grow. This is the Lasidioplodia theobroma, that after 4 days, it fill up entire the petri dishes, while some fungicide, in some fungicide, the fungi grow much less, but some of them they did not grow at all. From here, we took only those 5 fungicides that they prohibited 100% of the growth of mycelium, so which they were fine, and you can see that they are highlighted here. So we know these 5, they are effective on growing fungi in the lab, but we wanted to test them on the trees. Again, first we tested in the greenhouse, and the method was the same for inoculation. We inoculated the fungi, and then the same day we applied the different fungi, those 5 fungicides. And you can see in this graph that all 5 fungicides, they produce the lesion. Their lesion was significantly shorter than when we didn't use the fungicide. This is the trees that we did not use the fungicide, and this is the length of the length of the lesion that it was produced. And here are Tia and Tilt, they really produce a shorter lesion. So now we know that the fungi are effective, but they are in greenhouse, because greenhouse is a controlled condition. We tested in the field and see how it works. One of our farmers generously let me test on her 1 year, on his 1-year-old trees, which we applied the same methods, but we did in a greenhouse here. And after 7 weeks here, we saw 4 fungicides. They were most effective. And as you can see, this is when we didn't use any fungicide. The length of the lesion were longer than the other 2. Now, for the last step of this project, I really wanted to know if how this fungicide going to be effective when the trees have a dieback in the farm. So first, we did a very pilot study on 30 trees, and the results was promising. But we had to test it on more trees, and one of the farmers in Nursery Hills had 100 trees in his farm that they had the dieback. So in December, what we did, we cut the trees and then sprayed them with those 5 fungicides and with drainages. So far, by today, he says the dieback has not come back yet, but end of this month, we're going to do the second application of the fungicide and see after 2-3 months if dieback come back or not, hopefully not. So for the other part of the project, we were so curious, where is this fungi that we found, Botrytisperia and Dieportis, they are coming from to the orchards. For that, we first start collecting some, visiting some nurseries and then collecting the samples. The samples were taken from the very healthy, and you can see here, collecting from the healthy trees. And the results that we got, it was very interesting. We found different species of the Botrytisperiaceae. When we compared with those species that we found them on dieback, one species, which is the Macadamia, was common in both trees from orchards and nurseries. Then the second one was that I wanted to find out how about those plants that are growing around the macadamia orchards, and what species of Botrytisperiaceae do they have it? So we collected the samples from 10 different, most commonly growing plants around the macadamia orchards, and the results again was very interesting. And we had 2 species of that we found it on ficus and eucalyptus. They were found on macadamia trees too. So in conclusion for this project, we found 9 species that they are associating and causing dieback, but 1 species is the most virulent. And in artificial inoculation, NELMAC2 and NELMAC4, they are producing a shorter lesion compared to bound. And so far, the 4 fungicides are Thiamira-Bismuth, Luna Experience, and Tilt are quite promising. And 4 species we found from the nurseries, which only 1 species was common between both nurseries and orchards. And 3 species of Botrytisperiaceae, we found them on the surrounding trees, which 2 of them were common between orchards and surrounding trees. So we don't know if they have. I think we know that this macadamia, Dieportis macadamia, it comes from the nursery to the orchard. And these are, I just have to mention for those one that you don't know, these fungi are actually endophyte. It means that you saw that in the nursery trees, they were very healthy, but they had this fungi. But as soon as they have the trees or hosts, they are under stress, especially in the orchards, this fungi can be problematic and cause a disease. So I would like to thank Elia, Scott, and other colleagues that they helped this project running, and thanks for listening.
Are there any questions for Fahimi? Elria, you first again.
Yeah, sorry, I think this is important and it's not a question, it's just more of a disclaimer to everyone listening. Please remember that this is a research project. It's for research purposes. This is not technical advice, and many of those products are not registered for use on macadamias. So just that disclaimer, this is a research project.
Thank you, Aria. I think that's an important point. There's a second question. I don't have a name for it. Aria, Tian, you second.
That's me, Schalk.
Yeah, sorry,
yes,
man. My rekenaar wys nou jou naam.
Right. I just want to mention or ask something. You know that propiconazole and cyproconazole will be discontinued in June. So you have to look for something else again. Some other trials.
Actually, I talked to Syngenta and because they gave the chemical and I talked to them and they have registered propiconazole until next 10 years. That is inside information.
Okay, that's good.
They can continue.
And cyproconazole?
No, only propiconazoles. That's I've heard from them.
Okay, thank you.
Amy, that's very, very good news. Robert, you second in line.
Hi there, it's actually Karen from Fairview
Hi,
Farm.
Karen.
Hi there. I'd just like to find out the application of the fungicide. Was it on the soil or as a foliar application?
Hi, Karen. Thanks for your question. Yes, we in a greenhouse, they applied as a spray and in a small trees in the farm also in the spray. But I didn't mention that one, but I did a small pilot study also and we did a drainage and spray. It wasn't very significantly different when they produced the lesion. So for orchards, because spraying is not really feasible, and so we use the drainage.
Aria, new hand.
Oh, no, sorry, old hand.
No problem. Any more questions for Fahimi? Fahimi, if I may, I'm going to ask you a question just from my observations in the field. And I think I know the answer, but I think it's important to know. When you walk in the field, I see more than one type of branch or Bacillus for the lack of a better word, symptom. You do get the odd branch that dies completely and then can then be pruned back. But I also see a more general type of dieback. It starts with the tips of the trees. And sometimes even where the trees adjoin, it looks to me like, adjoining trees can transmit the disease from one tree to the next. Is that also your experience?
Yes, Schalk, not only macadamia, in any trees that they have a dieback. If the dieback branches does not cut in time, the fungi produce the spores on the dead branches because wants to survive. And with the water splash, wind, everything, it moved to the neighboring trees. That's why you usually see the neighboring trees start having a dieback. So when I'm in the farm, I always tell the farmers they have to cut the dieback branches and remove it from farm. Yeah, they have to burn it, discard it. Yeah, burn it is the best. They should not leave it in the orchards. And also sanitizing the pruning tools is very important. Because it is an endophyte, they might not see it, but it can transfer it easily to one tree to another trees. I don't know if I answered your question.
You absolutely did, and that is basically why with this kind of disease you see it spreading down a line of trees. Normally, if the guys didn't prune the equipment properly, I guess.
Exactly.
Any more questions? There's more. Tian, you.
That's me again. I just want to ask you something. Yeah. We've seen that when they do the harvesting, they use bamboo. And they hit these branches, and these two pathogens are wound pathogens. Isn't that the entry point of these fungi to enter the branches?
Yes, Tian, actually it is. Wounds are entry point, but usually that's what I've heard from some farmer. They said immediately after when they harvest it, they prune it and spray it. That is, I don't know if everyone does that.
More questions for Fahimi? Dion, thanks for that. That was actually a very important point that you raised. If there's no more questions, the talks are recorded, and if someone wants to listen, we will. Yeah, it is recorded. If there's no more questions, I'd like to thank our speakers, Fahimi and Gerda, who is not here, unfortunately. And then for everyone who attended, I think this is the way we're going to sort things out by training and by transferring knowledge as good as possible. And thanks for attending, and keep a lookout for the second one. We're going to advertise it in due course. And it will probably be related to plant quarantine pests and how to deal with plant quarantine. For everyone that has to deal with Macadamia felted cosset, I think this is hopefully a very important topic. And with that, I wish you a pleasant afternoon. Thanks, everyone. Goodbye.